Meiosis only accounts for about 10% of the actual cell cycle. The remaining 90% is called interphase, which consists of 3 steps. The first step is called G1, where the cell physically grows. Then comes synthesis, where DNA coils come together. The last step is G2, where the cell does the remaining amount of growing that it needs to do before entering meiosis. However, most cells are actually in the G0 phase, where they're non-dividing. Interphase also consists of 3 major checkpoints. These include the G1 phase checkpoint, G2 phase checkpoint, and M phase checkpoint. These checkpoints consist of molecular signals telling the cell to either keep dividing or stop. G1 is the most important because it checks whether the cell has enough nutrients to continue throughout the whole cell cycle. If it gets the okay to keep going, it usually completes the whole cycle. If not, it enters the G0 phase where it's non-dividing.
A cell that enters meiosis begins the process by coiling its newly duplicated DNA into chromosomes. As you can see, this cell has 2 homologous pairs of chromosomes, 1 pair from the mother and 1 pair from the father. A normal cell at this stage would have 46 chromosomes, with each half from each parent, but we're following one section of DNA for the sake of simplicity. This way, after dividing 4 times, each cell has 1 chromatid for each of the 23 chromosome DNA sections from the parents.
Meiosis is the same thing as mitosis, but instead of 4 phases, it has 8. In the first phase, prophase, the chromosomes are in the nucleus while the centrosomes are on the outside.
Crossing over occurs in prophase 1. In crossing over, one homolog, the bottom part of the chromosome, gets exchanged with the DNA from another homolog. This exchanges a part of DNA from one parent with the DNA from another parent. This creates an increase in genetic variation. All of these crosses are random and are not favored. So, it might cross over and it might not. The crisscross regions are called in chimaster form, which holds the homologs together until anaphase 1, where they are split apart by spindles.
In the next phase, metaphase, the centrosomes go to opposite ends of the cell, creating spindles. Next is anaphase. The chromosomes split apart and the centrosomes move even further to the opposite end of the cell, stretching it. The chromosomes are lined up. In the last step, telophase and cytokinesis, 2 cells form, still connected, which is a haploid. There is 1 nucleus in each cell with 1 centrosome. Inside the nucleus is chromosomes that are crossed genetically. This process starts all over again twice, 1 from each haploid part. However, this time the chromosomes are already crossed. In the final stage, telophase 2 and cytokinesis, 2 haploid cells are formed altogether.
Haploids only contain 1 complete set of chromosomes, and the most common example being gametes, or sex cells. Haploids are produced through the process of meiosis and actually make up diploid cells. This happens when haploid cells from the parent are fertilized. Haploid cells are made up of 23 chromosomes for humans, where diploids are made up of a complete set of 46 chromosomes. Diploids, unlike haploids, produce through mitosis and are what actually make up our muscles, nerves, skin, hair, and everything else that is not a sex cell.
In the end, 4 daughter cells are created from the original 1 of the parent cell. And because of crossing over, the DNA of the daughter cells will be different from those of the parent cells. Different DNA will then code for different genes, resulting in different traits in the organism.